In this video, we will discuss about the in vitro GlyTR1 results on triple negative breast cancer cells. So first, they have optimized their GlyTR1, which is this, its design is based on BITE concept. That is, it has this L-PHA lectin, which is bind through this linker L3, and this L-PHA lectin can bind to the another antibody, that is CD3 single chain fragment variable, and which is attached to this histag. So in this case, the CD3 will target the T cells and L-PHA lectin will target the beta-1,6 branching. The idea is to target this beta-1,6 branching on the cancer cells, and CD3 targeting on the T cells. And this is their antibody structures. Anti-CD3, they took the heavy chain light chain from anti-CD3 and the L-PHA lectin, and they combined together to make a GlyTR. And their idea is to use this GlyTR1 to make T cells and cancer cells close to each other, so that the T cells will get activated and kill the cancer cells through the release of lytic granules. So lytic granules are generally not released by T cells, but it might kill the cancer cells through the CD8 plus T cell cytotoxicity interactions. First, they checked their BITE productions, how they are formed, whether they are formed in the form of monomer, dimer, or multimer. They checked it using the size exclusion chromatography, and they are able to see that most of their BITE is formed in the dimer. And they validated it using the GlyTR1. They can see that the dimer formations are there, clearly evident from silver staining and the Western blot. And they checked whether the dimer will work best or monomer of this GlyTR1 works best. In supplementary results, they have shown that dimer works best in terms of the T cells activations. So they proceed with this dimer form of this GlyTR1. First, they check this GlyTR1 binding on different conditions of this primary T cells. On the control, they are able to see the binding, and in the MGAT1 knockdown, which essentially break down this N-glycosylation, but it does not affect beta-1,6 branching, so they do not see too much decrease in the signal. But when they do the CD3 knockdown, they are able to see a decrease in the signal, and when they did CD3 knockdown and MGAT1 knockdown, the signals is also low because CD3 is already knocked down. Then they wanted to check this, since the beta-1,6 branching are also present on the T cells, they wanted to check whether if they reduce this beta-1,6 branching, whether they are able to see some binding or not. So in the CD3 positive cells, Jurkat cells, they checked it with this kind of kifunensine negative and kifunensine positive. Kifunensine positive will indirectly affect the beta-1,6 branching. So when they use this kifunensine positive, they are able to see a reduced signal, but signal is higher than the background. And the background, they checked it using the CD3 negative Jurkat cells with kifunensine, so that it does not have CD3 and it also does not have beta-1,6 branching. So their signals with the CD3 positive and kifunensine positive is higher than the case where they have CD3 negative and kifunensine positive, indicating that the GlyTR1 can interact with the CD3 on the T cells. Now they check the GlyTR1 binding in the monomer and dimer conditions. The dimers, the bindings are significantly higher compared to the monomer. This is another evidence why they select the dimers. Now they did the co-culture assay where they took the PBMCs from the donors and cultured them with this triple negative breast cancers along with this different concentration of GlyTR1. They are able to see that when the GlyTR1 increases, concentration increases, the cell death increases, indicating the GlyTR1 can bring the T cells in the PBMCs close to these triple negative breast cancer cells. Next, they checked it. So one of the possibilities in this case is that CD8 plus T cells might also be active. So in order to check the CD8 plus independent cytotoxicity, which is mainly because of this GlyTR1, so what they did is they first checked the binding, GlyTR1 binding with the MSC1 deficient triple negative breast cancer. MSC1 deficient cells will not interact with CD8 plus T cells. So here they first check this GlyTR1 binding with MSC1 deficient TNBC cells. They are able to see that binding is high, and the control they use this B2, which has this MGAT5 knockdown. MGAT5 knockdown will not have this beta 1,6 branching, so in that case they do not see any binding. This is a negative control, indicating that the bindings are because of this beta 1,6 branching interactions with this GlyTR1. Next they check the cell death. The cell death also they are able to see the cell death where the based on this GlyTR1 binding in the not because of CD8 plus T cell cytotoxicity, because these cells are deficient of MSC1 and MSC1 is required for the CD8 plus interactions to these cancer cells. So this cell death is because of this GlyTR1 interactions with these cancer cells and the T cells in these PBMCs. Here they use this 2:1 effector to target ratio. Next they checked whether their killing because of this GlyTR1 can activate the immune cells or not. So they checked it by checking the activated CD8 plus T cell. It is CD8 plus T cells and CD25 plus T cells. They are able to see that when the cell death is when they co-cultured the CD8 PBMCs with these cancer cells, they are able to see, along with this GlyTR1, they are able to see increase in the CD8 plus activated CD8 plus T cells, compared to the case where they don't co-culture them with cancer cells, indicating that this activation, this GlyTR1 dependent cell killing can also help in the activations of the immune systems. So this is what they shown in this first figure, where they check the GlyTR1 in vitro efficiencies in terms of the cell death and cell binding. So that's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.